Yeah. All right. Good morning, everyone. I'm Noé Fernández Pozo from the Institute of Subtropical and Mediterranean Horticulture in Malaga, Spain. And today I will present EasyGDB, that is a tool we have developed to facilitate the implementation of genomic portals and expression atlases. First of all, I would like to thank the BCB for giving me the chance to present my work here in this seminar, and also for the excellent work they are doing in the network along this year. And with that, we can start. Sorry, I have to click in the screen. Yeah. So EasyGDB is a tool that is quite easy to install. You can install it in Mac, in Windows, or in Linux. You can find the code in GitHub. And also we provide Docker containers for Mac and Windows. And it's also quite easy to install in Linux. If you want to install it in a server, for example, it's basically based on PHP, and it doesn't have many dependencies. This tool was already published by Aureliano Bombarelli and I in 2022 in the journal Bioinformatics. And it provides a genomics portal that is ready to deploy. Basically, when you install it, you already have a genomics portal with all the tools and sample data for all these tools. And basically, the only thing you have to do is just to start customizing your site and start adding your own data. And for that, we provide a documentation where everything is described step by step and with example for all the data, so you can see the format and just replace it by your own data. Also, the main idea of EasyGDB is to simplify the data management. One of the things we have done in the latest version, for example, is we have removed all the dependencies for any relational database, because we realized that for many of the users, for people that only have basic bioinformatics skills and not so expert in SQL or relational databases, that was the main bottleneck for them. And now everything works without any relational database. It's as efficient as before, but you don't need as much time to update any data, and it's more accessible to many more people, because many more people can do these changes without the need to have very advanced bioinformatics skills. EasyGDB is very useful to manage and develop multiple genomics portals simultaneously. That's basically the main reason because I developed EasyGDB was quite selfish. It was because at some point I was involved in so many projects that required this kind of tools that I needed for myself a system where I could control all of them in a single place and implement it in a simple way and minimize the time I have to spend in updating the data or making any change that I needed. So basically now with a single configuration path file, you can just comment out all the projects but the one that you want to have active, and that way you can change between the projects that you have. It's quite easy. And as a piece of advice, anytime that you want to create a new project, you can just copy the example folder and just rename it by your own project name and just start replacing the data and customizing your sites. Also, the data upload in EasyGDB is quite simple. We tried that most of the tools, basically, you just need to place your data files inside the data folders specific for each one of the tools, and automatically everything will work in the tools. You will have access to this data set in the input page and also in the output for all the results. So you don't need to do much more than that. Also, EasyGDB is quite easy to customize. It is a modular structure, so basically all the tools are independent of each other, but they are also interconnected between them. And with a single configuration file, you can set up the path to all the tools. But by default, everything will work in Docker and also in a server. We have several variables that will be helpful also to customize, for example, the site logo, the name of the site, to limit the inputs and to control many other things. And something very interesting is like we can control the tools that we want to enable or disable, like switches. So basically it's like we have variables for each one of these tools, and you can just turn them to 0 or to 1 if you want to disable or enable any of these tools. And that way you will have them accessible in the toolbar. Or otherwise, if you want, you can create your custom toolbar, and you can just link all the tools that you want to have. And additionally, any tool, if it requires extra customization because we want to have more control of this tool, we provide a JSON file for each one of the tools where you can control all these changes. EasyGDB, basically, when you install it, it provides a set of basic tools that we would like to have in any genomics portals. Tools like many of you will know, like BLAST, search for gene ID or functional annotation keywords, file downloading. We have a genome browser based on JBrowse. We can have a Symphony viewer based on JBrowse 2. We have also a gene expression atlas, which is a tool that is quite complete and have many visualization methods. We provide tools also where you can provide a list of genes, and then you can extract, for example, the sequences for these genes. It could be hundreds of genes or thousands of genes. Then you can use these sequences for further analysis. The same way you can provide lists of genes and extract all the annotations for these links. Sometimes, for example, you get results for some experiment, like, for example, differential expression analysis, and you only get the gene identifiers. With that, it will be very easy to add all the annotations for these identifiers. We are also developing tools for passport and phenotype navigation and search for germplasm banks. We are developing also more tools for genetic variation, for gene co-expression, for gene ID lookup, in case that you want to have multiple versions of gene annotation and you want to have which genes correspond to the other versions. So you can use that between different databases, like which of your identifiers correspond to the ensemble or NCBI or for orthology, things like that. And also we have a tool for gene set enrichment. As an example of the tools that you can find in EasyGDB, because we have the basic file downloading that is quite simple to use. You just have a downloads folder where you just place the folders and files you want to provide in your genomics portal, and the system will replicate what you provide in the download folder in the download section. It's as simple as that, and you don't need to set up a FTP site or anything else. We also have BLAST that everyone knows and is quite useful. It has some features like you can provide nucleotide or amino acid sequences and they will be automatically detected. To add databases is quite simple. You just need to add the BLAST databases in the folder called BLAST-DBs, and that way they will appear automatically in the input of BLAST. Also, you can categorize them in subfolders, so you will have an extra drop-down menu here if you want to classify them by, I don't know, by type of sequence, by species, by databases, any data set you have. And the output that it provides is like the classical output at NCBI with a graphical color showing the score. We also have the possibility to download the results in a tabular format. We added also an extra information here with the identity percentage. And below you can find a table summarizing the results, and even below you can find alignments and more information. Also, the links for the genes will take you to the information of the genes in the genomics portal, but also if you have a genomic sequencing that you are querying, you can link that to the genome browser, or if you have a data set from an external database, like, I don't know, identifiers from UniProt or anything else, you can also provide links in a JSON file to any other database. The thing is, how BLAST works in EasyGDB is like everything works in the RAM memory. So the input file and the input sequence and the output is all stored in the RAM memory. So it doesn't create any temporary file, doesn't write anything on the disk. So that later you don't need to maintain anything. You don't need to remove any temporary file. So it makes things easier and doesn't require any maintenance. Other example of tools, for example, for gene annotation search or for the extraction of annotation from a list of genes. Basically, you just need to provide a file, a tab delimited file with your gene identifiers and the identifiers in other databases and the functional description in these other databases. And automatically, you just can use all these tools. These tools will provide, as a result, a table and many other tools also in some of the result required tables. And for that, we are using jQuery data tables, and we are trying to implement many features on them, and they are common to all these results. So all the results that you get, you can download in different formats. Again, they will be linked to the information of the genes. In this case, we will provide all the annotations with the identifiers of the databases with links to these external databases and the functional description. You can search within this table, you can sort them, you can change the order of the columns. There will be different pages to organize the results if there are thousands of them. And that is like a standard for all the tables in the results of EasyGDB. Also, as I mentioned before, like the results from BLAST, from the annotation search and other tools will take you to the gene information page. These are dynamic pages created with all the genes, and for that they use like the information in the genome browser, the annotation file, and the databases that are in BLAST. And that way we can create dynamic pages for thousands of genes, so you can find all the functional annotations, the sequences of these genes, and also the information of these genes in the genomic context. Also, one of the most interesting tools that we have, I think this one is quite complete, and you can install it independently of the others, is the Gene Expression Atlas. So in many cases, we have projects where we just have created a Gene Expression Atlas without all the part of the genomic portals. It is very easy to upload the data. As I showed before, you just need to provide the file and you will get it. It provides multiple gene comparison in many of the visualization methods. It is very interactive and highly customizable. For that, we have 7 different visualization methods, and you can download the expression data together with the annotation. And it is compatible with many different omics type. So you can use RNA-seq data, you can use expression microarrays, proteomics, metabolomics. And I will show you an example of all the visualization methods that we have in the Gene Expression Atlas. First, as the input is, as I showed you before, is if you just place your data file in the expression data folder. This is just like a count matrix, a normalized matrix with the replicates. It could be in any units. Automatically, once it is in the folder, you will be able to see it in the input, and automatically the code will read this file and will provide you like a placeholder the first 5 genes of the file, and automatically will assist the users to autocomplete the name of the genes in case they want to find any gene and add it to the list, or you can just paste a list and get the expression for these genes. Also, if you want, again, you can organize the input in different subfolders, so you will have different categories. In this case, I made an example with 2 species, and you will have an extra drop-down menu where you can select between the different species, and then below you have the different data sets available for that species. And as you change between the dataset, you will get here automatically the gene names, in this case, for example, for olive, and in the other example for mango. And that is something that you don't have to set up anything to get access to that. Just place your matrix of data in the data expression folder. Also, one of the first visualization method that we provide in the Gene Expression Atlas is the expression images or cartoons. And here we have some examples of the cartoons that we have done in other projects. All these cartoons are done by different people, and it was not really too complicated for them to create them. To show you how you can create these cartoons, so you can get an idea, it's like some people have used BioRender, other people use other basic tools for drawing. Or you can use even like a Keynote or PowerPoint. The only thing you need to get at the end is basically like images that have like a transparent background. In this case, this gray background represents a transparent background. And then you draw just the sample with a white color, and the white color will be replaced by the expression color, like the 2 images that you see here. So the code will just take these images that you create and overlap one on top of each other. And if they don't overlap, they will just create these nice cartoon images for this representation, and then they will have the color expression. And just with a JSON file, you just need to say, okay, this image corresponds to this sample, this image corresponds to the sample, and automatically you will have the cartoon ready to use. Then we have other visualization methods like line plots or bar plots. In this case, you can see the the gene expression for multiple genes simultaneously, so you can compare the expression of these genes in this representation with lines. Sometimes if some lines are hiding the values of others, or the ranges of expression are quite different, you can click on the name of the genes and the scale will readjust and will hide the genes that you don't want to see. You can select any of them and you can hide or show the genes that you want. If you mouse over the genes, you will see this gene highlighted. So it's quite easy to compare multiple genes simultaneously and adjust it to the scale that you need for the genes you are comparing. The same way we have the bars. You can just switch, clicking here, and you have a similar representation, but in a bar plot. And again, you can hide or show the genes. You can download all these visualization methods in different formats. We have also a heat map visualization. And again, in this heat map visualization, you can compare the gene expression of multiple genes simultaneously in multiple samples. This heat map visualization provides different color palettes and also is highly interactive. You can go to mouse over any of the ranges of expression, and then it will highlight all the samples that are within that range. We have also another visualization method to show the real pictures of the phenotype. For example, in these expression cards, where the cards will give you in the frame the color of expression, you will have the value of expression, and you can actually see how the sample looks like with a real picture. With sentences is much better than just a sample name, because that way you can actually see, for example, what is how bad is a disease in a fruit or in a sample. Or here, for example, we have the development of the pollen. You can actually see what is the growth of the pollen tube at each one of these hours. It's not only the time that you have in the information of the name, but you also have an actual picture that is giving you more information about the sample. We also provide a visualization method to see the replicates. So you can see the expression of each one of the replicates of all samples, and that way you can see if there is a high dispersion between the replicates you're checking, or if they are clustering together very well. So that way will allow you to evaluate if that sample is more or less reliable. And finally, the last method to provide the results is a table that summarizes the expression data for the genes that you have selected and the samples of the data set. Here again, you can download all this data. You will have links to all the genes. You will have the average values of the samples. And together with that, you will get all the annotations for these genes together with a link to external databases. Also, if you want to install your own expression atlas, this is quite customizable. So we have in the configuration file I showed at the beginning, you can set up, if you want, what is the order of the visualization methods that you want to have in the atlas. You can enable or disable them, or in this case, for example, at the top, we provide usually the information about the experiment, where we have in the publication of the experiment, what are the conditions, the samples, where can you find the raw data, and information that you want to provide about that. Then later, we have the visualization methods like the expression images, line and bar plots, the expression cards, the heat map, the replicates, and the average table. But if we want to customize that and we want to change it, for example, we can disable all the tools that we don't want to show, and we can change the order. And for example, we can say, okay, first I would like to have the table, then in second place the heat map, and at the end I would like to see the replicate. So then it will create a visualization like that with the table at the top, then the heat map, and then the replicate. Also, another tool that we have developed for EasyGDB is like a navigation and search tool for passport and phenotype for a GERMPLUS collection, because that is quite interesting in our institute, because we are hosting one of the most important collection of subtropical species in Europe for many species, especially for cherimoya, avocado, and mango. And for that, we have created a system where we can navigate through all the species that we have. We can explore all the accession of the collection that we have in the map to see the origin of this species. And of course, when we go to each one of the species, we can visualize each one of the accessions in a table with a lot of information. So before we have a large collection of phenotypical and passport data collected for many years, and we wanted to provide a system that was more visual and provide to this data not only just the numerical or categorical data, but also pictures about the information of these traits. So for example, what we have done is we are using guides of descriptors for this species. In some cases, we also create our own pictures. And that way, for each trait, we just provide what the pictures of all the possible values that this trait can get. For example, in avocado, these are all the possible colors that the avocados usually can get. So that way, with this set of images, we can provide like a visualization guide for all the accessions that we have. And we can create dynamic pages that will provide for each trait a set of images that will represent what is the categorical value of this. And for example, here, for the accession fuerte, we have that is medium green. And instead of just having this name here, medium green, we also have these images that is telling you, okay, these are all the possible colors, and this is yours. And in the case of another accession called Maluma, it's the same, but this is tell you this one is black, so it's this one. That way, we have created a passport and phenotype tool. And I will show you an example of how it looks like for avocado, for one accession, for Maluma. So at the top, you can see a summary of the passport information. It provides a QR code in case you want to print it and have it in a tree or in exposition or in any information that will lead you to this page, and you can see all the information. We have a section with a gallery where you can provide pictures of the fruit or any part of the plant or any organism that you want to have here in this collection with real pictures of this accession. We have also created a section that summarizes what are the most important traits for this species, for example. This is called feature descriptors. And for example, in avocado, then we would like to know, okay, what type of inflorescence it have? If the flowers open in the morning or in the afternoon, because this is very important for pollination. Is the ripe fruit, is it black or is it green? Is the surface of the fruit, is it smooth? Is it rough? And any kind of traits that are interesting for your species, you can just summarize it here to have it very fast at the top. And also we provide a map with the location of the origin of this accession. In this case, it's from South Africa. And later we have all the phenotype tools. In the phenotype tools, here we have also the possibility to add other languages. So in this case, it's in English and also in Spanish. We also have the possibility when we have numerical values, we provide the average, and also we have a category within a range based on the guides of this phenotyping data. And if you click in the information button, you can get an extra information with all these ranges. So you can tell really your measurements in which category they are and what are the other categories. So you can know more or less what are the ranges available in the whole collection of avocados. Also, in many cases, when you are characterizing a collection with a phenotype, in this example, for example, you can take 30 different fruits, and not all the fruits from the same accession always have the same value for the same trait. So if you want to do an average, then some cases it's not always the same. So in this case, for example, what is the color of the layer next to the skin of the ripe fruit? So in 28 cases, it was medium green, and in 2 cases, it was light green. So now we're providing a summary of all values that were found here. So you can have between parentheses the actual number. And at the end, we have a table with all the replicates that were collected with all the details of each one of the traits. And you can see here all the ones that were medium green and light green. And more or less, this is how it looks like when you navigate through the phenotype for the ripe fruit descriptors of avocado in our collection. If we have like 100 accessions of avocado, then these dynamic pages will be created for the 100 accessions. And this is only for the ripe fruit, but we also have that for the inflorescence, for the leaves, also for the fruit at the time of harvest. And we are also developing this for cherry mola and for mango. And it's quite simple, more or less, to create for for germplasm banks and other sites that need this kind of collections. And if you don't have many phenotype data, you can create that very fast. And it's much more visual than just providing a table with text. Also, something very nice once you have this tool is that you can have some search more complex that will help you to design experiments for GWAS, for RNSEC, or even to know which one of the accessions are more interesting for some traits or others, for example. Here, I decided to search between different of the phenotype data, and I say, okay, I want to know which are the avocados that have fruits with a length equal or longer to 10 centimeters, and the surface of the fruit is medium rough or very rough. And the color is not green, it's black, medium purple, or reddish. And then once we perform this search, it will summarize all the information that we search in the collection, and it will tell you all the accessions that meet all these conditions. And below, of course, you can access to all the phenotypical data and passport data following the links or in the tables that we provide. And with that, I would like to finish. I didn't want to make this presentation much longer, so we can have any discussion later if you want in the questions. So I would like to show you some examples of the implementations of EasyGDB we have so far in a couple of years. So we have now created AboBase, Chickpea 10K, which is a genomic portal in collaboration with the Murdoch University for more than 10,000 genomes of chickpea. We have also MangoBase, Olive Atlas, which is a expression atlas for olive. We have also skeletal atlas for human and mouse. We have the IGSM subtropicals, Horcrux base, Madland expression, Ethionema arabicum expression, and a few more. And with that, I would like to thank all the people that participated in this project, all the collaborators that work in these platforms, all the funding agencies, and also my group here at the Institute. So thank you very much all for your attention.